each participant, the kit will include 1 disposable transfer pipette, 4 blood spot cards, and 1 no additive vacutainer. You will also need access to a biohazard waste bin. To avoid clotting, begin immediately after completing the blood draw, and begin setting up your workspace. Open the transfer pipette and lay out the blood spot cards, taking care not to touch the 5 dotted circles on each card. Remove the cap from the vacutainer. Using the transfer pipette, draw up some fresh blood. Carefully dispense 1 drop of blood into each circle on the card. Continue this process until all circles on all cards have been filled. Once complete, place the cards in a safe location and allow them to dry for at least 3 hours. After they have been set aside to dry, recap and dispose of the vacutainer with the remaining blood. Also dispose of the used transfer pipette. Finally, we'll review a few common mistakes to avoid. Each circle should be filled with a single drop of blood. Avoid underfilling or overfilling. Do not touch the pipette tip to the card surface, and do not add multiple drops to the same circle. Do not blot, smear, or dab the cards with a tissue. Once the blood has been added, avoid touching or handling the blood spot card until they are dry. Okay, I can also send this video, hopefully to upload to Box as well, if that would be helpful. But here we just have an example of a properly collected blood specimen. So you can see there's 1 drop in each of the circles. There's no overfill. There's no blotting. You know, we can't be perfect with all of these, but that's why we're getting 4 cards from every participant. And then each participant's card will have their identifier on it and then the date that it was collected. And then here is just some information showing invalid specimen collection. So you can see these have dried out, they've bled over, they haven't been allowed to dry for the whole time. Some of these have, you know, missed the dot completely, underfilled. So all just super helpful information, but we're going to try to make it super easy for you guys and provide all the information and all of the tools that you'll need to be able to collect the blood spot cards. And I will pass it to Chun to go over the why of the blood spot cards.
Yeah, so a few people have asked why we're doing blood spot cards in addition to the actual blood draws themselves as well. The reason really is that, you know, in addition to identifying the biomarkers, we'd actually like to see whether we can perhaps find a less invasive way to monitor those biomarkers. So the idea really is that we'll use the kind of larger volume of blood that we collect to actually do the biomarker discovery and the validations. And once we've actually found some candidates, we would actually go back to the blood spot cards and look to see whether we can detect those changes in the blood spot cards. And if we are able to, then it would demonstrate that we could potentially use blood spot cards in the future for, you know, say, you know, either monitoring disease progression, you know, rather than doing a blood draw, we could just do blood spot cards, which would make it kind of less invasive and easier for some people. Or, you know, potentially, you know, if the biomarkers actually show up early enough in the disease course, you know, you could potentially even look at doing newborn screening with those. So really the idea with the blood spot cards is not necessarily to do the actual discovery work, but just to kind of validate to see whether we can detect them in a different medium that could be used in future trials and research. Any questions? Nick?
Thanks. Would you be willing to accept dried blood spot cards that are done with finger prick? And then is there a minimum necessary number of DBS cards for those that would potentially be only able to be a finger stick?
I think we're asking for the protocol. I think we're asking for 4 blood spot cards. I don't see, and Joel, correct me if I'm wrong, but I don't see any issue with doing a direct fingerprint collection versus
Nick, I see no issue with it. It's really just to determine whether whether the matrix that we're collecting in a blood spot card could be used for some of these mass spec based assays that we read from straight from blood. And then as Chun mentioned, either for ease of collection going forward, could we use a finger prick? That would be ideal, yes. But are some of these biosignature biomarkers feasible enough that we could get a good enough robust reading out of a blood spot card to use for newborn screening down the road as well. So I would be totally fine with finger pricks.
What kind of volume do we expect it to take to fill up for complete cards?
I think in the past when we've done this on whole blood cards, we usually need no more than 1 ml to 2 ml tops to be able to do 4 cards.
Each dot is recommended for like 50 microliters if you're using an actual calibrated pipette. So 50 times.
Yeah, so a ml would be more than enough.
Mm-hmm.
Are there any other questions from the group? Oh, Erica.
I'm wondering what are the best practices around leaving the blood spot cards to dry for 3 hours? And I also am curious if that potentially raises any challenges for folks in terms of, do they have a space to protect the specimens for that amount of time? But can you give us some guidance on a general approach to how to think about that? And then if anyone has any challenges with that, also please let us know and we can explore that a little bit.
Yeah, when we get them, that's a great question, Erica. When we do these, we have like a chuck pad that we lay down. So we can provide those as well. And then we just find a private space in the lab or in an office at the conference when we've collected just in the side where there's not an air vent nearby, where there's not a lot of direct sunlight. You don't want to dry them with like a hair dryer or anything. You want to try to avoid all of that. And a place where there's not a lot of maybe dust falling down as well. And we just stack them up, with all the blood hanging out so it can air dry, and then just let them chill for 3 hours at least. But
and if you needed to walk away and didn't want them to be disturbed or even seen, you could lay a sterile chuck over the top of them or one of the pads over the top of them as well. They could also go into a lab drawer or file drawer or cabinet to dry. It's really just not to disturb them and not to spill anything on them or get anything on them or have direct air blowing on them.
So they could be contained in some way if need be.
Yes.
So for each site, that will be something to think about. Where and how will you allocate space in accordance with whatever your unit or floor institutional guidance is for where samples and biospecimens can be left potentially, well, likely unattended. What does that look like for your unit? And that'll be something to think through. Are there any other questions?
I have a question about the collection. Is this something for cohort 2 or cohort 1? Will parents potentially be doing this at home?
In general, and actually, Jill, why don't you take that?
Yeah, in general, these would be collected at the same time that the other blood draw collections are done, Suzette.
Okay.
So like, as you know, with our protocol, we do send out collection kits to families as well, independently for Project One. We have not historically, when we do those, done blood spot cards, just because we know they are a little bit more complex for a family member to do them. So we've historically only done these at the family conference and collected them that way. But our hope is that going forward, as a part of the main protocol, when someone gets a blood draw, they would also do the blood spot cards in parallel.
Okay, yeah, we attempted first, and we sent them back to Boston. Yeah, and it kind of was a nightmare. I'm not great at that stuff anyway. But the whole prick in the circle and, I mean, I have a tough time drawing within the circle of taking one of those tests. So anyway, yeah, it was, but it would be great if we could have something at some point that could be easier to do that for sure, but okay, thank you.
Well, if there are questions that come up, please feel free to add them to the chat as we go, or to reach out after the meeting and we can make sure those are addressed. I want to thank the whole Sanford team for a really fantastic presentation. It was just very clear and easy to understand. And Joelle, I think that video is going to be a very nice reference for when people actually go back to do this for the first time. Very much appreciate the do's and don'ts as well. So thank you, everyone.